Hello friends, in this video I'm going to take this topic of protein separation methods or protein purification methods, and that is chromatography and electrophoresis, by the help of which we can separate the proteins. For example, we have a mixture of proteins, we want to separate the individual proteins, so we can use these two techniques, chromatography and electrophoresis. Now usually students don't understand these topics at this level because you have never done these experiments, chromatography and electrophoresis. Once you do the experiments, things are so easy. So right now when I'm going to take the lecture, in a form of experimental form, I will be taking this topic in diagrammatic form so that you can actually see what is happening. So question solving will be very easy for you, right? So let's start. First, I'm going to take chromatography. So let's say I'm giving you a mixture of proteins. This is a flask where I'm giving you a mixture of proteins. Now how will you separate? First you will analyze the mixture. And here when you are analyzing the proteins, you can see the difference is in size or molecular weight. The difference is in size or molecular weight. So when we know that the difference is in size or molecular weight, then we will take a column. In the column, this is a column taken. In the column, we will take gel molecules. The size of the gel is such that the smaller protein can enter the gel, but bigger protein cannot enter the gel, right? Now we will pour the mixture on the top of this column. When we pour the mixture on the top of this column, then we have both big and small protein here, and both will be coming down with gravity. But there's a difference. That smaller protein will enter the gel, then leave, then enter, then leave, then enter. Like this, smaller is coming down. But bigger protein will be coming down from in between spaces because this protein cannot enter the gel. So can I say that smaller protein is taking time to come down, right? Because it is getting hindrance from the gel. Smaller protein is taking time to come down, but bigger protein is coming down first. Bigger protein eluted first. We use the word eluted or elution, right? That is coming out first, and after some time smaller protein will also be coming down. So here we can separate the two proteins. First we will be collecting the bigger protein in a different test tube, and after some time when smaller protein is coming down, then we can collect it in a different test tube. So we have separated the mixture of proteins. Now this kind of chromatography is known as gel filtration chromatography. That is, we are using a gel. We are doing a kind of filtration and we are using gel for filtration called gel filtration chromatography. Now they can ask you that which size protein is coming down first, is eluted first in case of gel filtration chromatography. You can see here larger size, bigger size protein will be coming down first. In a normal filtration, what we do in a normal filtration, the smaller thing is, the bigger thing is upward and smaller thing is coming down, right? So here it is opposite. Bigger protein is coming down first and smaller will be coming later, right? Also, this chromatography is called size exclusion chromatography because we are excluding them on the basis of their size, right? Now, the gel which is used in gel filtration chromatography, commercial name of that gel is Cefadex. When they ask you these questions, they just write Cefadex as one of the option. So you have to know everything behind this Cefadex word, which is commercial name of this gel. Biochemically, this Cefadex is dextran, D-E-X-T-R-A-N, dextran, which is another homopolysaccharide made up of alpha glucose, high molecular weight structure. This dextran is a plasma volume expander in patients of hypovolemic shock, right? This dextran is also used for making gel in gel filtration chromatography, right? So we're done with gel filtration chromatography, which is one type of chromatography. Let's take another kind of experiment in this case. Again, I'm giving you a flask, and in this flask, I'm giving you a mixture of proteins. So first is you will analyze the mixture, and on analysis you came to know that the difference is in charge. So when we know that the difference is in charge, we will take some charged molecule in the column. Again, I'm taking a column. in the column I am taking CMC. I'm just showing one line of CMC just to make diagram easy, but otherwise in 3D picture this column is filled with CMC. What is a CMC? This is carboxy methyl cellulose. Carboxy means negative charge, amino means positive charge. This I told you in basics. So carboxy is negative charge. So we have negative charge in the column. We have negative charge in the column. Now we will pour the mixture on the top of this column. So we have both positive charge and negative charge proteins here. Now the column is having negative charge. So the positively charged proteins will be attracted towards CMC and they will stay in the column. The positive charge will stay in the column, but negative charge will be repelled out. Negative charge will be repelled out. So eluted first. Negatively charged protein is eluted first. This way we have separated this mixture of proteins, and this chromatography is known as ion exchange chromatography. That is, ions are getting exchanged, right? If we take negative charge in the column, negative charge protein is eluted first. So if we will take positive charge in the column, then positively charged protein will be eluted first, right? Now third such kind of experiment. Now I am giving you a flask in which I am giving you a mixture of two proteins. So on analysis, you came to know that one of the protein is an enzyme. One of the protein is enzyme. So again, we will take a column. In the column, we will take coenzyme. We will take coenzyme. Enzyme has affinity for coenzyme. So when we pour the mixture on the top of this column, we have both proteins, enzyme and other protein. Enzyme will stay in the column because it is having affinity for coenzyme, which is lying in the column. On the other hand, other protein is eluted first. This kind of chromatography is known as affinity chromatography. Known as affinity chromatography. Affinity chromatography can be used to separate enzymes, antigens, hormones, vitamins, antibodies. So we can take any compound which is having affinity for the one present in the mixture, right? And any compound having affinity for each other, that affinity thing can be used for separation here, so called affinity chromatography. So we are done with these three types of chromatographies: gel filtration chromatography, ion exchange chromatography, and affinity chromatography. These all three are actually types of column chromatographies. All are types of column chromatography. So can I say column chromatography can be based on three properties: size or molecular weight. In biochemistry, consider size and molecular weight one and the same thing. It can be based on charge, it can be based on affinity. So this question was asked, column chromatography can be based on which properties, right? Now, in chromatography, one more thing we have to do, that which is mobile phase and which is stationary phase. In this case, what is the stationary phase in all these experiments which we have done? There's a common stationary phase. There's a common thing in all these experiments which is not moving, which is like this, staying, not moving. So that is column. Column is the common stationary phase in all these experiments which we have done. And what is the mobile phase? Which thing is moving down the column? Which thing is moving with gravity? Mixture of proteins, right? So the mixture to be separated, that is the mobile phase. Mixture to be separated is the mobile phase here, and stationary phase is column here. It is very easy to tell the stationary phase because name of chromatography tells you the stationary phase. When it is column chromatography, then column is the stationary phase. When it is paper chromatography, if it is paper chromatography, then the stationary phase is paper. If it is thin layer chromatography, that is TLC, then the stationary phase is thin layer of silica is used. Thin layer of silica is used. So name of chromatography tells the stationary phase. The name is having the stationary phase. So like this. Now, so these three types of chromatographies we have: column chromatography, paper chromatography, and TLC. We are done with column chromatography. Now these two are left. These two, paper chromatography and TLC, they have same principle. The principle is same, just that the difference is paper chromatography is the older but the cheaper version. It is less costly. But TLC is the latest, the new one, and it is the costly version of paper chromatography. Paper chromatography is mainly used for teaching purposes, but for research and diagnosis, we are using TLC, right? So let's see this common principle of paper chromatography and TLC. Now, first we will take the stationary phase, either paper or thin layer of silica, whatever. So we are taking the stationary phase, paper, and then we apply amino acid on one side. We apply amino acid on one side, and on the other side we make a line. And now we will take a beaker. In this beaker, we will take a solvent. This solvent which I am going to take is methyl cellulose media. This ethyl, methyl, aliphatic things, they are always non-polar. So I will say that this is a non-polar medium. We will dip the paper in the solvent. Dip the paper in the solvent with the side of amino acid attached downwards. On the other hand, a line is there, right? Now when we dip the paper in the solvent, then this solvent will move upwards with capillary action. The solvent is moving upwards. The paper is getting wet. When we are doing the experiment, we can see the paper is getting wet. The wetness is coming up. When the wetness has reached till this line, then we will take out the paper and we will do the staining of this paper. So let's say the results of my staining are: this amino acid has given a stain here, this amino acid has given a stain here, and this amino acid has given a stain here. So now, what is our interpretation? See here, what is the mobile phase? Which thing is moving upwards? Mobile phase here is not only solvent, but mixture of amino acid and solvent is moving upwards, because when solvent is moving upwards, it will also take along the amino acids which are present on the way. So mixture of amino acid and solvent is moving upwards. In this mixture, if see, the basic thing is a polar thing is soluble in polar solvent, a non-polar thing is soluble in non-polar solvent. So in this mixture, if amino acid is soluble in the solvent, then it will go along with the solvent till the last point. But if some amino acid is not soluble, then it will leave the solvent very early and will get attached onto the paper. So a very early stain means amino acid's polarity is opposite to the medium, and a very late stain means amino acid's polarity is same like the medium. So in this case, this amino acid is non-polar, this amino acid is polar, because this amino acid is going with the solvent till the last point. There is a late stain here for this amino acid, right? We know that solvent has moved till the last line, right? So some amino acid is giving a stain later till the near to the last line. Then that amino acid is soluble in the solvent, so that is non-polar here in this case. But this amino acid, that is polar because it is giving a very early stain here, right? See, if the medium taken is polar, then everything will be changed, okay? So mobile phase is mixture of amino acid and solvent, and stationary phase is paper or thin layer of silica, right? So we're done with chromatography. Now coming to electrophoresis. Now electrophoresis is movement in electric field. Phoresis means movement, electro means electric field. It is movement in electric field. For example, let's say this is electrophoretic plate. One side negative, one side positive. So let's say we apply mixture of proteins here in the center. So when we will switch on the electric field, then some proteins will move to this side, some proteins will move to this side. So movement in electrophoresis depends on? Yes, it depends on charge. Charge is the main factor on which movement depends. But this movement in electrophoresis also depends on size. If something is very heavy, it will not move. And it also depends on shape, that the shape is globular or fibrous. So that will also change the electrophoretic movement. So a normal electrophoresis depends on these three factors: charge, size and shape. But there is one special electrophoresis, SDS page. SDS page depends only on size. Normal electrophoresis depends on charge, size and shape, but this SDS page, very special electrophoresis, frequently asked topic in questions. So this SDS page depends only on size. The full form of page is polyacrylamide gel electrophoresis, and SDS is sodium dodecyl sulfate. SDS, sodium dodecyl sulfate. It is a salt derivative of lauric acid. A salt derivative of lauric acid. Lauric acid is a 12 carbon acid. Even dodecyl word means 12 carbon. Now, for understanding that how this electrophoresis depends only on size, we have to see the properties of SDS. Properties of SDS. First is SDS will denature the proteins. It will denature the proteins. When proteins are denatured, then secondary, tertiary and quaternary structures are lost. Now shape is mainly because of tertiary structure, so shape is lost. Right? Let's say earlier some protein was globular, some protein was fibrous. Their movement in electrophoresis was differing due to their difference of shape. But now they all have same shape. You can say they all are in their primary structure. The shape is not there, right? It's like destroyed. So this electrophoresis does not depend on shape, right? Second point about SDS is that SDS is anionic detergent. Anion means negative charge. Now let's say some of my protein is positive, some of my protein is negative. So they are moving towards different ends of the electrode. But when I will apply SDS, SDS will coat all the protein with negative charge. So now all the proteins are negatively charged. So the difference of movement of this electrophoresis does not depend on the difference of their charge, because all are having now same charge, that is negative charge, right? Now third point about SDS is that approximately 1.4 gram SDS will bind to 1 gram of protein. So for example, a small protein, less SDS binds. Bigger protein, more SDS binds. The difference of size is still there, but the difference of shape and charge is gone. That's why this electrophoresis only and only depends on size of the proteins. Now there's one more fourth point about SDS, that SDS cannot break disulfide bond, but disulfide bond is present in tertiary structure of the protein, and in this experiment we have to break disulfide bond. But SDS cannot break disulfide bond, so we can use some other compound to break disulfide bond. See here, this is disulfide bond. We can add either oxygen and we can break, or we can add hydrogen and we can break, right? This is called oxidation of disulfide bond. This is called reduction of disulfide bond. The chemical which oxidizes disulfide bond is performic acid. The chemical which reduces disulfide bond is mercaptoethanol. So in this experiment SDS page, in addition to SDS, we will either take performic acid or we take mercaptoethanol to break the disulfide bond, because SDS cannot break disulfide bond, right? Now there's one more special electrophoresis called isoelectric focusing. We will take this, but first let's take the basics. You know, amino acid structure is this, that there is carboxy group on this side, amino group on this side, and H here and R here. But in body, amino acid gets ionized. It will get ionized to give negative charge on carboxy and positive charge on amino group. So this kind of structure, net charge is 0. Net charge on this structure is 0. Actually, solubility depends on charge. Solubility of amino acid or protein depends on charge. When there is no charge on the structure, then this kind of structure is insoluble. So when the structure is insoluble, then precipitation will occur at this point. And this kind of structure of amino acid having negative on carboxy, positive on amino is called Zwitter ion. So Zwitter ion kind of structure in amino acid will cause precipitation here. There is a term Pi. Pi. Pi is that pH at which Zwitter ion exists. So can I say that at Pi a protein will precipitate? Because insolubility will occur here because the net charge is 0 here, right? Now let's see what is isoelectric focusing. That is, using isoelectric pH, we can cause the separation of proteins by electrophoresis. See how? Let's say this is an electrophoretic plate. In this electrophoretic plate, I am taking different pH in different areas. Here pH is 3, here pH is 9. We are taking different pH in different areas in an electrophoretic plate, and one side is negative, other side is positive. Now let's apply mixture of two proteins here on this side. Both proteins are negatively charged. So when I will switch on the electric field, then these proteins will start moving towards positive end of the electrode, right? Now, one of our protein is having its Pi of 3. So, when this protein will reach this area, it will be insoluble and will cause precipitation. That protein will be precipitated in this region, but other protein will keep moving further. Other protein is having its Pi of 9. So, that protein will precipitate here, right? So, this way, our two proteins are separated in different regions. And this is called isoelectric focusing. That is, using isoelectric pH, we have focused the proteins in various different regions and we have caused the separation here, right? This is it with this topic. Thanks for watching.